Dear friends and colleagues, it's my pleasure now to invite you to the last session of this fascinating symposium. The session is entitled Oncology, Hematology and Tumor Immunology, 3 areas that go perfectly together. And we have 2 excellent tandems with presentations. The first tandem is Myriam Gyrospe from the National Institutes of Health in Baltimore, USA. And the tandem partner is Steffen Fuchs at the Department of Pediatric Oncology and Hematology at the Charité. In order to save time, I would like to start immediately with the session without a long discussion of the speakers, and I would give the word to Myriam Gyrospe to start.
Thank you very much. Good afternoon, everyone. I would like to begin by thanking the organizers for the opportunity to join this fantastic program, and also for the chance to provide an introduction and provide context for the studies that my colleague, Dr. Steffen Fuchs, will present afterwards. So we both share a keen interest in circular RNA biology. In the case of my laboratory, our interest is in the process of aging. And so we are interested both in the normal decline of aging, as you'll recognize here, loss of muscle mass, loss of vision, hearing, immune function, etc., as well as the diseases that arise as we age, the pathologies, many of which have been discussed at this conference, neurodegeneration, cardiovascular disease, obesity, diabetes, cancer, etc. Now, we study these declines and diseases of aging by focusing on cell model systems. And we do this because one of the best documented changes that occur as we age is the ability of our cells to respond to stimuli. So if we take cells from a young person and we expose them to growth factors, hormones, DNA damage, oxidants, nutritional imbalances, etc., those cells respond very well. And the tissue preserves homeostasis, and that leads to a maintenance of health. But if we take cells from an older person, those responses are impaired. And over time, that impairment leads to loss of homeostasis and the onset of disease. So understanding how cells respond to stimuli has been really an area of focus in my lab for more than 2 decades, and has given us great insight into the biology of aging. A major component of this response are changes in gene expression programs. And just to bring everyone on the same page, this is a busy slide, but I just wanted to illustrate the fact that gene expression programs are regulated on many levels. Transcription, of course, leading to the production of a pre-messenger RNA, and a host of post-transcriptional processes that lead to splicing of the mRNA, export, degradation, transport, storage, translation, etc., in a way that ensures that the right amount of protein is made in the cell at the end, in the right tissue, in the right moment, in response to the right stimulus, etc. These transcriptional and post-transcriptional steps are tightly regulated by factors that bind RNA as well as DNA, including proteins and non-coding RNAs, among which I have only highlighted 2 of them here because I will refer to them in my talk. MicroRNAs, which are 22 nucleotide long RNAs that typically repress gene expression by lowering mRNA stability and reducing translation, and circular RNAs, which is the topic of this talk, and as well as the following talk. Circular RNAs affect gene expression on multiple levels, and I'd like to offer 3 examples of such levels. Together, non-coding RNAs and RBPs, or RNA binding proteins, elicit this very tight and very dynamic and versatile regulation of gene expression in the cell. So what I'd like to do in the remaining minutes of my talk, I'd like to give you a little bit more introduction about circular RNAs, and then offer 3 examples of how circular RNAs affect gene expression program using specific examples from my lab work that we've done in my group. So circular RNAs have been known for many decades, but really they have come to the forefront of RNA biology in the past decade. And Jeremy Willis and Phil Sharp have been among the pioneers, which have taken advantage of the fact that RNA sequencing had revealed vast families in diversity and number of circular RNAs in every mammalian cell. So in a very typical way of thinking of circular RNAs is the result of a pre-messenger RNA, which instead of being spliced into a normal matured mRNA that exits the nucleus and goes to the cytoplasm, the circular RNA formed by recircularizing exons or back splicing, and then very often they are found in the cytoplasm. But as you can see here, they share sequences with messenger RNAs. And given this feature, they can bind certain factors that would otherwise be bound by the messenger RNA. And this was true and shown first for microRNAs. So the idea that circular RNAs can sponge microRNAs was very attractive and has led to a lot of research. We know that circular RNAs can form from single exons as well as from exon-intron combinations. They may also be just intronic, etc. So they're very diverse, very abundant in the cell. They exist in tens of thousands in each mammalian cell, and they differ depending on tissue. We also know that the function of circular RNAs is very often tied to the factors that circular RNA binds. I mentioned microRNAs already, but they may also bind long non-coding RNAs, messenger RNAs, DNA. They may bind a host of proteins, RNA binding proteins or RBPs, transcription factors. They may even bind ribosomes. And resulting from this interaction, some circular RNAs, the exception, not all of them, some circular RNAs are partly translated, and the micropeptides that form from that translation have other biological functions. So, okay, so what I'd like to do today is, and these are 3 examples of interactions of circular RNAs with factors leading to changes in gene expression, as well as the translation I just mentioned. So I'd like to offer 3 examples that cover these 3 types of interactions with factors, beginning with one that focuses on the interaction of a circular RNA and microRNA. For this, the context that I'd like to introduce is one of the model systems that we study the most in my lab, and that is cell senescence. You don't have to look at all of them, but it's one of the traits, one of the changes that occur in our tissues with aging. You may recognize others here as well. We can study cell senescence by taking primary cells, such as fibroblasts, and allowing them to undergo replicative exhaustion. By shortening of telomeres, the cells stop dividing. And so Amaresh Panda, a former postdoc in my lab, asked a very simple question. He said, are there differences in circular RNA composition between proliferating cells and senescent cells? You might imagine we found many, many differences by RNA sequencing. And one of them, a circular RNA called SIRPVT1, was found to be lower in senescent cells. So that was interesting to us. And it was even more interesting when we realized that silencing SIRPVT1 alone, so just that intervention, triggers senescence. So we had proliferating cells. If we silence SIRPVT1, we began to see these senescent cells that we recognize because they turn blue due to the presence of a senescence-specific beta-galactosidase. So Amaresh went on and said, well, how might SIRPVT1 be eliciting senescence when it is ablated from cells? And so the first thing he did was focus on possible microRNAs with which SIRPVT1 might interact. And we predicted, so this is a prediction table, that all these microRNAs had binding sites on this circular RNA. But then he went on and did an experiment in which he pulled down SIRPVT1 from the cell and then asked what microRNAs are binding. And so to do this, he designed a biotinylated antisense oligomer that recognized the only unique sequence on the circular RNA, which is this junction sequence that does not exist in any linear RNA. And then by RT, reverse transcription, and quantitative PCR analysis, in the pull down, he found that actually only LED7 was enriched. So this was the microRNA with which SIRPVT1 was binding in senescent cells. And to make a long story short, what this led us to discover is that in proliferating cells, where SIRPVT1 is very abundant, LED7 is sequestered, is bound to this circular RNA, and taken away from other mRNAs that encode proliferative proteins. So these mRNAs can then be translated and cell proliferation occurs. By contrast, in senescent cells, SIRPVT1 levels are much lower. And so here's LED7 is free to bind target mRNAs, including many that encode proliferative proteins. And here we see growth arrest. Okay, so having told you about this example of a circular RNA binding this classic binder, microRNAs, I'd like to switch to circular RNA binding RBPs. And here, the example that I'm going to use is sort of focuses on one of our favorite proteins, HUR. This is a protein implicated in cancer, implicated in senescence, immune response, etc. And the question that Amaresh asked in this particular project was, can we identify the subset of circular RNAs to which HUR binds? And so we did we used a method called RIP for ribonucleoprotein immunoprecipitation that consists in the IP or immunoprecipitation of HUR, preserving this complex so that we can then, in the IP material, identify the circular RNAs. Of course, we always have a parallel IgG IP so that we can subtract background. And again, as you might imagine, Amaresh found many, many circular RNAs to which HUR was binding. He identified them using microarrays. And at the top of this list of enriched circular RNAs was an RNA with a number name for now that was very highly enriched in the HUR IP, not so in the IgG IP. And we renamed it to PABN1 because this circular RNA, in fact, originated from this precursor RNA, the PABN1 pre-mRNA, which gives rise to the circular RNA from a single exon, exon 6, I believe. And otherwise, the pre-mRNA is spliced into PABN1 mRNA, which encodes for the poly-A binding protein N1. So the very curious thing about this circular RNA is that when we look closely at all the sites where HUR might bind on this single exon of SIRPABN1, HUR was capable of binding across the body of the circular RNA. And this was really interesting because if we thought that this might be influencing HUR function, we now see that the circular RNA can bind HUR across the entire body of the circRNA. And moreover, since HUR multimerizes, forms oligomers, we can really envision how SIRPABN1 might be an effective sponge. And again, to leaving out a lot of the molecular specifics that Amaresh went on to show, the conclusion from this study was that if HUR is bound to the PABN1 mRNA, we see active expression of PABN1. But if alternatively, the circular RNA emerges from the precursor RNA, and I'll say in parentheses that we have no idea how circular RNAs are formed or what triggers their formation, what determines whether they're made or not. If the circular RNA is formed, then HUR is sequestered into the circular RNA and PABN1 is left without HUR and translation decreases. All right, so the last example that I wanted to touch on to illustrate all the exciting and unexpected things that circular RNAs can do focuses on transcription factors. And here the context that I'll offer is the fact that with advancing age, we lose muscle. Here's a 78-year-old man compared with a younger person, great muscle loss. And this, of course, is linked to frailty and loss of independence. So the process of muscle regeneration in the adult is of great interest in the Institute. And in my lab, we use a cell model of myogenesis, where we put myoblasts in culture, and we allow them to fuse into myotubes as it occurs in our muscles. And so Poonam Pandey, another former postdoc in the lab who was interested in this model system, asked whether circular RNAs might be contributing to the process of myogenesis. Again, conceptually a very simple question. So she extracted RNA from myoblasts, myotubes, and then looked for enrichment in myotubes versus myoblasts. And she found one specific circular RNA that was not just very abundant overall in myoblasts, but also highly overexpressed in the differentiated myoblast GM compared with the growing myoblast GM. SIRC74, a little bit more about it, is 519 bases long, and it originates from this precursor RNA again, single exon, exon number 2, and this precursor RNA would otherwise give rise to 74, which expresses the protein 74, important for muscle physiology. So again, Poonam asked whether 674 first had a function in myogenesis. And to do this, she designed siRNAs in order to silence circular RNA. And once again, we focused on the junction sequence, the only sequence that is unique to circular RNAs. And you can see she was able to reduce SIRC74 levels, and that led to impairment of myogenesis in culture. So she asked, what factors might CXCR4 be binding to. And I will add that the circular RNA, of course, is more abundant in differentiated myotubes. You can see some of them here. If we zoom in, you can see that compared with the messenger RNA, which is in teal color, the circular RNA is present in the nucleus as well as sometimes in the cytosol. So she asked, what is CXCR4 binding to? And she did this again by designing an antisense oligomer with which she could pull down the circular RNA and any proteins bound to it. So in this case, we focused on the proteins, and to keep the search wide, she used proteomics. And so 2 specific proteins were found enriched in the CXCR4 pull down, per beta and per alpha. Others as well, but this caught our attention because it was found in both human and mouse myoblasts when we repeated this experiment, and also because this protein has a really important function in myogenesis. Per alpha and per beta are repressors of the myosin heavy chain gene. And again, to leave out a lot of a detailed analysis that Poonam did, what this revealed to us is that in proliferating myoblasts, because the levels of this circular RNA are so low, at least in part, per alpha and per beta are repressing myosin heavy chain production. But as myogenesis advances and CXCR4 levels rise, per alpha and per beta are sequestered away from the promoter, so they no longer repress myosin heavy chain production, and now myosin heavy chain can be transcribed and protein can be produced from this gene. All right, so I'll just wrap up by telling you that I've offered 3 examples of circular RNAs, different in each case in different contexts of aging, that interact with microRNAs as well as with RNA binding proteins and transcription factors. And by doing this, these circular RNAs help to elicit certain gene expression changes. So I thank my lab. This is before the pandemic. We cannot be so close up anymore. And Poonam has left my lab. He's now finishing her postdoc at MD Anderson, and Amaresh now heads his own research lab at the Institute of Life Sciences in India. I'll thank you all for your attention and invite Dr. Fuchs to the podium. Thanks.
Yes, thank you so much. We will move on to the Dr. Fuchs, Steffen Fuchs, who is also working on circular RNAs. There's a million of questions we could have, but I guess we discuss them after both of the talks.
Yeah, thank you for the kind introduction. And I will tell you now something about my clinician scientist project, which is about the landscape of circular RNAs in neuroblastoma. And I'm especially happy today to speak together with Myriam Gyrospe, since we already met 2 years ago in person, actually, at the circular RNA course at the EMBL laboratory in Heidelberg. Yes, this was obviously also before the pandemic. Okay, neuroblastoma is my topic. It's a malignant tumor of the early childhood. It's an embryonal tumor which is derived from precursor cells of the neural crest. So that means this tumor can basically arise everywhere in the body where you have peripheral nerves later on. But most of the times we find tumor masses in the abdomen, as you can see here, the MRI. So in Germany, we have around 130 cases per year, and most of our patients are very young, so younger than 1 year. Problematic is that half of those patients already present with a high-risk constellation at the point of diagnosis, and this greatly limits their prognosis. So you can see here in the Kaplan-Meier that the 5-year overall survival is below 40%. We don't see so many genetic aberrations, but what we see in around 20% of the cases is an amplification of the MYC and oncogene, which is routinely diagnosed in the DNA fish, as you can see here. Now we learned almost everything now about circular RNA biology, and I would like to take the chance now to bring this back to oncology, since Myriam told you circular RNAs can regulate gene expression on different levels. One can imagine really easily that if this gets disrupted, this can also lead to cancer. And I would like to use neuroblastoma as a model disease now to explain this. We got interested in circular RNAs in neuroblastoma because the lab of Nicolas Rajeski from Berlin, they conducted a study where they analyzed basically circular RNA expression in different tissues of the body, and they saw that it was highest abundant in neural tissues, as you're seeing in the cortex. Further, they differentiated precursor cells of neurons, so neuroblasts, and they saw that circular RNAs were differentially expressed, if even tightly regulated. So when we come back to neuroblastoma as an embryonal tumor that could not properly differentiate, one could also easily imagine that if circRNAs were dysfunctionally expressed, this could also promote neuroblastoma formation. So for our study, we analyzed 104 neuroblastoma patients, took their primary tumor samples, analyzed them by total RNA sequencing, and bioinformatically determined circRNAs, linear RNAs, and characterized candidates. And by this means, we could identify more than 5,000 circRNAs from 2,000 genes. And here you see the number of genes that produced circRNAs, and it was then evident to us that actually most of the genes, they did not produce circRNAs. But if they did, then they only produced a few isoforms. So before this circRNA renaissance, a lot of people, they used to believe that circRNAs, they were just byproducts of mRNA production. But this would actually mean that they should have like a positive or almost perfect correlation with mRNA expression. That's why we also did this analysis. So we took the mRNA expression and the circRNA expression over all our tumor samples and analyzed the correlation. And we saw actually a very low correlation, indicating maybe an independent biogenesis of circRNAs. And further, we looked also at the genome-wide level where circRNAs were produced in the genome. And in yellow, you see here the tracks of the mRNAs where they produced in the genome, in blue the circRNAs, and then we calculated the ratio of circRNA over mRNA. And wherever you see a peak, that means there's a hotspot where more circRNA is produced than mRNA. So for instance, you have this on chromosome 20 or also on chromosome 14. Another indication for an independent mechanism of circRNA production. We then dug deeper into the different neuroblastoma risk groups, and you see this here. So basically, we differ between high-risk neuroblastoma samples that have or have not a MECAN amplification, intermediate risk group, and then also low-risk neuroblastoma. And then we wanted to understand what is actually the global picture of circRNA expression in neuroblastoma. So we took the expression of circRNAs globally in the neuroblastoma samples, performed a hierarchical clustering, and then we found 3 principal clusters. And quite interestingly, in this cluster 2, we found almost all MECAN amplified high-risk neuroblastoma samples in brown here. So that means they have a distinct expression of circRNAs. We also had a deeper look in clinical characteristics of our patients, so overall survival events, event-free survival events, and so on. But what I want to highlight here is actually the number of circRNA isoforms and the circRNA expression in the different risk groups and the patient samples. So red means a high number or a high expression in these categories. And then you can already see that actually for the MECAN amplified samples, it seems to be a bit low. And in panel B, we summarize this in the box plots. And now you see it quite clear that actually the MECAN amplified samples, they have the lowest amount of circular RNA isoforms. And in C also you see that the expression was especially low. So very, very interesting for us that these high-risk samples have the lowest expression. So that's why we decided now to take only these high-risk neuroblastoma with or without MECAN amplification, compare them. And in panel A, you see the differentially expressed genes. So we see MECAN and associated genes upregulated, also several MECAN, so published MECAN targets were up and down regulated, and also a lot of splice factors affected. And then when we take now the circRNAs and calculate the ratio of circRNA over mRNA per sample and per gene, and do this globally. So you see here the cumulative distribution in brown for the MECAN amplified samples and in orange for the high-risk non-MECAN amplified samples. And the more left you go in this graph, the lower the ratio gets, and what the y-axis gives you the probability. So you see that the ratio gets especially low for the MECAN amplified samples, and this is significant. Of course, you can now ask the question, tumors are heterogeneous. Is this really a MECAN effect, or is it biased by something else? That's why we decided to take now a cell line system that can express MECAN by induction, so by tetracycline. And you see this in panel C. We took this neuroblastoma cell line and used MECAN expression for 5 days. And you see already the picture is more homogeneous. But again, we could induce MECAN, and we see also a similar effect on MECAN targets and also splice factors. And when we then take, again, the ratio of circRNAs and linear RNAs, calculate the ratio globally, then you see here for the induced samples in red, this ratio becomes especially low, and this is also significant. And then finally, we integrated those 2 data sets, and then we see that the overlap is very, very high. So that means means two things actually. The cell line model could reproduce the findings in the tumors, and that actually it's now very likely that the effect is MYC-N driven. But how does this exactly happen? One indicator could be RNA binding proteins, since they were, for instance, reported to regulate circRNA function in biogenesis. So that's why in the next part, we took the RNA binding proteins that we detected in our tumor sequencing data and correlated their expression with the expression of circular RNAs and performed a hierarchical clustering analysis. And you see this here. And this brought us basically two principal RNA binding protein clusters and two circular RNA clusters. And blue means here an especially low correlation. And you see here in this cluster 1 circRNAs, they have an overall very low correlation. So we asked ourselves, could we maybe find here in this cluster all the downregulated circRNAs from the MYC-N amplified samples? And we integrate those data sets, and then we see this here in this Venn diagram that the overlap is really large. So almost all the circRNAs that we found downregulated in the MYC-N amplified neuroblastoma are also found in this cluster. And now these RNA binding proteins that have the weak correlation with those circs could be negative effectors. So we had to look at this list and compared it with the literature. And here we saw one RNA helicase, which is called DHX9, that had a low correlation of only 0.2. And was already described as being a negative regulator of circRNAs. So we wondered, was it regulated by MYC-N, and is it also effective in neuroblastoma? We'd look at the DHX9 expression on the RNA level in our high-risk neuroblastoma RNA sequencing. We saw it was overexpressed in the MYC-N amplified samples. And when we looked at published mass spec data, then we saw also that DHX9 on the protein level was also upregulated. Further, we looked at published MYC-N ChIP-seq data in 3 MYC-N amplified neuroblastoma cell lines, and we see here a very nice peak in the DHX9 promoter, another indication for a MYC-N regulation. And finally, we took our inducible MYC-N expression system, induced MYC-N expression for 2 days, which you see here in the Western blot, and then we found also DHX9 was upregulated, another indication for this MYC-N regulation. So we were confident that we could also find such a negative regulation now in our cell line system. So we performed the knockdown of DHX9 by siRNA and sequenced it. And you see in the MA plot a great shift of circRNAs. So globally, most of the circRNAs were upregulated after DHX9 knockdown. When we calculate then again the circRNA over mRNA ratio, then we see the other way around as before. This ratio becomes now especially low for the control and not for the knockdown. So that means the knockdown puts this ratio in a positive way, so upregulates circRNAs. And then when we finally integrate the DHX9 knockdown dataset with the RNA-seq from the MYC-N amplified tumors, we see an almost perfect overlap. So that means DHX9 could be one of the main effectors that drives the circRNA repression in the MYC-N amplified samples. In the next part, we were interested in circRNAs that were specifically upregulated in neuroblastoma. And for this end, we compared our RNA-seq data with data sets that were published on different adult cancers and also pediatric cancers and controlled it with healthy brain tissue. And you see here in green, we found 26 circRNAs that were upregulated in neuroblastoma. And among those, I will show you now some data of a circRNA derived from the ARID1A tumor suppressor gene. This is especially interesting for us because it is mutated in around 11% of neuroblastoma and then also associated with an unfavorable prognosis. You see in panel A, the ARID1A gene, which gives rise to a circRNA derived from 3 exons here. And we have again this characteristic backsplash junction between exon 4 and 2. We amplify this by PCR, and you see here the Sanger sequencing of this junction. We could also directly detect the circRNA by RNA FISH, here clearly in the cytoplasm of the neuroblastoma cells. To further understand what circARID was doing in neuroblastoma cells, we performed the knockdown, which did not affect ARID1A mRNA, neither the protein. And we could already see that, for instance, caspase-3 levels decreased, indicating apoptosis. We also saw that the cells were dying under the microscope. We saw that the cell viability was decreased in an ATP-dependent cell viability assay. We also saw, for instance, that proliferation was decreased. When we had a closer look at their phenotype, we saw that here control versus knockdown, they also tend to have a more differentiated phenotype. You see more outgrowth and network formation. But how does this happen? So as we already heard from Myriam, circRNAs affect their function mostly by interacting with other molecules. So we also performed a pull down of circARYT1A, and we're focusing now in interacting proteins. So we performed a mass spec of those enriched protein fraction, could identify a lot of binders, and among those were the ELF-L proteins and also KHSP, important RNA binding proteins that affect, for instance, RNA stability and also splicing. We confirmed the pull down by PCR, so we could enrich for circARYT1A, but not for the mRNA and other transcripts. And it was also reversely confirmed by an RNA IP when we take now antibodies for the interaction partners KHSP, ELF-L, we enrich for circARYT1A, but not for control transcript. But then we had a look actually in silico on the circARYT1A sequence to see if we could also predict those binding sites. And for ELF-L, we didn't find any. So this indicates maybe a more indirect interaction. But for KHSP, we find 3 sites, making KHSP the more interesting interaction partner for us. But how relevant is this interaction actually? Here we analyzed the KHSP expression and the circARYT1A expression in the tumor RNA-seq and analyzed the correlation, and we saw an overall positive correlation of almost 0.5. To further understand how relevant the interaction is also for KHSP, we performed a circARYT1A knockdown, which we could confirm by qPCR, and we saw that the KHSP protein was decreasing, while the mRNA stood stable. So that means actually that circARYT1A could have a stabilizing effect on the KHSP protein. And then we also wanted to understand actually what KHSP was doing in this context. So we performed a knockdown of it and sequenced it and did the same independently of circARYT1A. And then we integrated those data sets and looked at the differentially expressed genes. And you see now here in this Venn diagram that almost or even more than 50% of the genes found differentially expressed after circARYT1A knockdown were also found in a KHSP knockdown. So that means half of the changes found after circARYT1A knockdown could be found also in the KHSP knockdown. And then we also had a look at gene ontology terms, and after KHSP knockdown, we found, for instance, apoptosis upregulated and cell cycle downregulated. And this is actually what we've seen also after circARYT1A knockdown with our cells. So they were dying and not proliferating so much anymore. But was this also the case after KHSP knockdown in the cells? So we did this, confirmed it by qPCR and Western blot, and had a look at the cells. And also these cells, they proliferated less, and also the cell viability was decreased. So the same effects. So this means overall that KHSP could to some extent recapitulate the effects caused by a circARYT1A knockdown. And this brings me to the summary. So we identified more than 5,000 circRNAs in primary neuroblastoma samples. I showed you a repressive effect of MYC-N on circRNAs, which could at least be in part explained by DHX9. We identified neuroblastoma-specific circRNAs. Of those, circARYT1A is important for the neuroblastoma cells, and we identified KHSP as an important interaction partner of it. And this overall sheds light on the role of circRNAs in the pathogenesis of neuroblastoma. We're now working on validating those effects also in other MYC or MYC-N alterated tumors, and also trying to establish a circARYT1A knockout model. And finally, we want to evaluate the role of circRNAs as therapeutic targets and biomarkers in neuroblastoma patients. So I would like to thank you for your attention. I'd like to thank my mentors, Angelika Eggert and Johannes Schulze, and all the lab members that supported me, our cooperation partners, the funding buddies, and also, of course, the BIH and the Clinician Scientist Program, and Dushka Dragoon for the support over all these years. Thank you very much.
Okay, Dr. Fuchs, this was a matching talk to the first one by Myriam Gyrospe, really bringing into light what circular RNAs can do. There is the possibility to ask questions, which will be in the chat. While this is going on, I have one question, general question to you, I think to both of you, because a number of people in our audience are not so familiar with the phenomenon of circular RNAs. And these molecules have long been neglected or even overlooked because of the sequencing methods used to look for RNA species in cells. And the question is, how did you stumble over this class of molecules which were so long overlooked? And So it would be nice if you just tell a few things, how you got into this for the audience.
You want to start?
Sure. We have always been fascinated by RNAs in all areas of biology. So for us, it was very natural to when circular RNAs became so central to gene expression biology, we quickly turned to them, and it was a very natural transition from other non-coding RNAs to circular RNAs. That's my general answer. Stefan?
Yeah.
Yeah, so I can give a personal answer. So I remember actually I was at the MDC doing a master thesis project on microRNAs in colorectal cancer together with Ulrich Keilholz. And then I heard actually a presentation from Nikolaus Rajewski at that time about circular RNAs. It was the first time that I heard about this class of molecules, and it was really intriguing what they could do. And yeah, now this knowledge extended much more until now. And yeah, this is somehow, I ended up on this topic because I wanted to continue working on non-coding RNAs, and this was the next step for me.
Okay, thank you. That is nice. And the next question is, if you would have molecules to target a circular RNA, what would a cell likely do to compensate for this? Because this is something important to understand potential mechanisms of resistance to inhibiting circular RNAs function.
I can begin just in general. If we had a way of targeting specific circular RNAs, we might want to target those that confer resistance to, for example, a tumor or a metastatic tumor, or circular RNAs that, for example, enhance angiogenesis, something we might want to prevent. That would be the goal. So, based on these examples, we would target circular RNAs that have an adverse effect on a particular disease that we're trying to combat or intervene. Again, very general answer.
Yes, I agree. You have to carefully select your target. I mean, I think this is always the case. And then I think now, even if you think about RNAi therapies, so as siRNA-based approaches, now this is more possible than ever, since we have now new formulations to make RNAs more stable, to bring it into the human system, for instance. Yeah.
Okay. There's a question in the chat now. Do you think that circular RNAs are rather diseased or aging RNAs, or are they physiological regulation?
I can start again. So circular RNAs are highly tissue specific. So they are expressed in different parts of the body. And again, we know so little about how they are expressed and produced that there's a lot of work to be done there. And some of them are associated with normal physiology. Some are associated with specific diseases. There is a great deal to know. Now, that question is essentially unexplored at this moment.
Yeah, maybe I can add something to the physiological function. Actually, again, a study from the Rajewski lab, where they found actually a network of circRNA in the brain. And for instance, they proposed that this is important in neuronal function, and they disrupted it in a mouse, and then you could even see behavioral defects. So indicating like a really important physiological function. So I think they are not only disease related, so they also have important physiological cellular functions, yeah. But we know still not enough about them.
Okay, this is always nice if you don't know enough of this, because you can move on. And in the session, we also need to move on. Thank you so much for this exciting tandem talk. And we move to the next tandem with Itay Tirosh from the Weizmann Institute of Science and Damian Rieke from the Department of Hematology, Oncology and Cancer Immunology at the Charité. And I would call on Itay Tirosh to start this tandem.
Hi, thank you for the introduction and for the invitation to speak. Do you see my slides?
Yes, we do.
Okay, great. So my name is Itay Tirosh. I'm a systems biologist from the Weizmann Institute in Israel. And my lab is studying cellular heterogeneity in cancer. So what all of you know very well is that when we say that different patients have the same type of tumor, they actually have distinct tumors. Each tumor is a unique entity. And that's the basis for precision medicine, and it's widely appreciated. What is perhaps not as well appreciated, and is the focus of my lab, is the fact that within each individual tumor, there's still quite a lot of diversity. So any tumor is in fact an entire ecosystem with a diversity of cells. This includes the immune cells, the stromal cells, and of course the cancer cells themselves. And even if we focus on just any one of those components, like the cancer cells, there's a lot of diversity within it. So cancer cells could be different from one another because of genetics. Many of the mutations are found only in a subset of the cancer cells, and that defines distinct clones of cancer cells. And there's also a lot of epigenetic regulations. Cells can switch between different cellular states. In many cases, they mimic processes of differentiation. They interact with the environment and respond to that. So there's multiple reasons why the cells are very different from one another. And all of that could have a lot of implications. For example, some of the cells in the tumor might be more resistant to a specific treatment, and this could be the basis for why tumors so often recur, even after successful responses. And similarly, there's this model, and I think a lot of evidence for it, that at least in certain cancer types, there's a subpopulation of cells that we refer to as the cancer stem cells that is particularly important for the initiation of that tumor, as well as for its recurrence. So these are two subpopulations that we would really need to identify better and target better to eliminate residual disease. And similarly, metastasis, which is of course the main cause of death in most cancer types, is also not something that is generated by all of the cells in the tumor, but just few of the cells are sometimes capable of undergoing this complex process. Even when we talk about immunotherapies, not all of the immune cells are actively recognizing the tumor as foreign, only a small subpopulation. So there's a lot of significance for the differences between the cells in the same tumor, and we would like to understand all of these differences and explore the subpopulations of cells within the same individual tumor. But the problem with all of this is that in the cancer genomics field, what at least used to be the case until recently, is that we typically profiled the entire tumor just as a single sample. And basically what we see is just the average of all of the cells in it. This is what was done in the Cancer Genome Atlas, TCGA, and in most of the studies until recently. So about 8 years ago, when the technology became available, we shifted into single cell RNA sequencing, in which we now take the tumor, we divide it into individual cells, we profile them one by one, and we get a lot of information, the activity of basically all of the genes in each one of the cells. And then we can start to reconstruct this diversity and understand the different subpopulations and start to explore their functions. And we've been using this approach in a number of different cancer types, but our main focus has and continues to be in glioma. So let me show you a few examples from what we see in glioma, and I'll start with oligodendroglioma. So here you see the diversity. Each dot here is a cell, and these are only the cancer cells, only from one patient, MGH54, but we see very similar patterns in different patients with oligodendroglioma. So what you can clearly see is that the cells largely separate into two different groups. The ones on this side, these are the cells that resemble oligodendrocytes, both in their morphology, but also in their gene expression. They express many oligodendrocyte-specific genes. And on the other side, we have all of these cells that instead express a program that is more similar to astrocytes. So we have these two different components, but perhaps more importantly, there's also these cells in the middle. And the cells in the middle, they are at an intermediate cellular state between oligodendrocytes and astrocytes, but they also express this stemness program. And more specifically, when we compare these cancer cells to all of the cells in the brain, we find that they are most similar to neural progenitors, progenitors that in normal development would give rise to both neurons and to the glial cells. So that suggests that we have a population of progenitor cells that might give rise to both the oligodendrocytes, which are more common here, and to the astrocytes, which are less common. And the colors that you see here correspond to the cells that are actively cycling. So from the single cell data, we could easily identify for each cell, where is it along the cell cycle? And what we see is that all of the cycling cells are in these components that we refer to as the stem-like cells. So all of this is very consistent with the cancer stem cell model, suggesting that there's a subpopulation, about 5 to 10% of the cells. These are the cells that are driving the growth of the tumor. These are the most important cells, but they are constantly also differentiating and giving rise to oligodendrocyte-like cells, as well as some amount of astrocyte-like cells. Now, the amount of these cells varies between different tumors, and we see that this correlates with how aggressive the tumors are. So for example, in IDH mutant disease, this is oligodendroglioma as well as astrocytoma. These are less aggressive types of glioma. We tend to see a small amount of cells in this area. When we studied DIPG, diffuse intrinsic pointing glioma, which is a very aggressive pediatric glioma, we see a similar picture, but now a much higher fraction of these stem-like cells, consistent with a very aggressiveness, with the high aggressiveness of this disease. What we could also do in the IPG is to now isolate those cells, inject them into mice, and we see that these progenitor cells very easily give rise to tumors in mice. And the differentiated cells from this area, even when we inject 100 times more of these cells, we do not get tumors in mice. So indeed, these are the cells that are most important for this disease. When we talk about glioma, of course, the most common and the most aggressive glioma is glioblastoma. So let's switch now to glioblastoma. Here we see a more complicated picture. So rather than 3, now we have 4 different cellular states: oligodendrocytes progenitor, neural progenitor, astrocyte-like cells, and mesenchymal-like cells. So each corner corresponds to one cellular state. And you can see that many of the cells are kind of somewhere in the middle. And this is because, as I'll show you in a minute, the cells can all the time switch between those cellular states. So we have 4 different cellular states. And what you see by the colors here is that in all areas, some of the cells are proliferating. So this is not like the previous slides in which only one population proliferates. Here we have a lot of proliferation basically everywhere. When we now look at individual tumors, we see that each of them is composed of the same 4 elements, the same 4 types of cells, but in very different frequencies. So each tumor has a different composition, but they all contain the same types of elements, the same types of cells. So now we need to better understand what generates these different cellular states and what are their implications. So let me show you a few more things that we learned about this. First, we went back to some of the patients and we tried to now isolate individual states. So from the single cell data, we identified markers that allow us to isolate. For example, using CD44, we can isolate the mesenchymal-like cells. Using CD24, the NPC-like cells, or we can just get all of the cancer cells. And we now inject them into mice. And in all cases, we get tumors in the mice. And this, first of all, indicates that these multiple states are capable of initiating tumors. What was also quite interesting here is that the tumor that we now get in the mice, in the PDX sample, has the same composition, more or less, as what we saw originally in the patient sample. And this is true not only when we start from all of the cell types, even when we start from just one type of cells, the composition of the tumor in the mice is now similar to what we saw originally in the patient. And this and additional experiments demonstrate the high amount of plasticity. Cells can actively switch between one state and another. Now, this transition makes sense in some cases. Neural progenitors, it would make sense from a developmental perspective that they would turn into oligodendrocyte progenitor and to astrocytes. But one state was particularly intriguing. This is the mesenchymal-like cells, because we don't expect in glia development to see mesenchymal-like cells. And so we were trying to follow up on this and to better understand the origin of these cells. And what we noticed is that those tumors that have a large amount of these mesenchymal-like cells are also enriched with macrophages. We see more macrophages in these tumors, suggesting that there might be some form of communication in which the macrophages causes the cancer cells to adopt the state in which the cancer cells resemble mesenchymal cells. And I'm not going to go into the primary data for lack of time, but just to give you the bottom line, what we find is that macrophages tend to secrete a lot of cytokines. One of these is called oncostatin M, OSM, and the receptor for OSM is highly expressed on the glioblastoma cells. So this is the OSM receptor. The interaction between OSM and OSM receptor activates this mesenchymal-like program, which is shown here in pink. And then we get these tumors in which the more macrophages there are next to those tumor cells, the more mesenchymal cells we see in the tumor. And the other thing we learned about these mesenchymal cells is that they seem to be important for the interactions of the cancer cells also with the T cells, partially because they increase the expression of MHC molecules. So we are now trying to understand whether this mesenchymal-like program could be actually useful when we try to treat those patients with immunotherapies. So overall, I showed you that we see in each of the types of glioma that we study a lot of heterogeneity. An important point is that while each tumor is unique, the patterns of heterogeneity are quite similar between those different programs. So when I say that in IDH mutant disease, we see this pattern, that means that across many different patients, we always see the same components, the same populations of astrocyte-like cells, oligo-like cells, and stem-like cells across each patient. And the fact that we see the same pattern suggests that we need to incorporate this in how we treat those patients. So in one type of hierarchy, we see one stem-like cells, and these are the cells that we need to target. In the other example that I showed you, in glioblastoma, we have multiple populations that are all important, and then we probably need to come up with some sort of combination treatments to be able to eliminate all of these states. So finally, I want to thank everyone in my lab and everyone that worked on this project. All of the work that we do in glioma is a very close collaboration with Mario Suva and his lab at Massachusetts General Hospital. And thank you for listening.
Thank you so much for this fascinating talk. There is many things we could discuss, and I would love to discuss with you at this point. But in order to really keep the tandem alive, I would like first to ask Damian Rieke to give his presentation, and then we can have the discussion together of both talks.
Thank you very much for having me, and thank you very much for some great talks already and for being here. It's an honor to speak among such amazing or guest speakers. My name is Damian Rieke. I work for the Department of Hematology and Oncology at the campus Benjamin Franklin, and I broadly titled my talk, Improving Personalized Cancer Therapy. I tried to bridge the gap between the audience that is not familiar with cancer therapy and those that are specializing in it, to try to give you a broad overview, but also to go into some details. I first want to start off and show you a background of where we are in precision oncology. I want to give you an idea of what the problems might be with our current approaches, and also some ideas of where the field might be able to go, followed by a summary. To give you a background, in precision oncology, we are oftentimes faced with a patient population where we do not really well know what to do. Those patients have advanced cancers, they are pre-treated, they have diverse cancers, and basically we start with nothing for those patients. And then we start with sequencing, which is in itself a very complex process. And I just want to briefly bring you through this slide to give you an idea of what we have to deal with. First of all, naturally, the sequencing process of the tumor material itself, the data production. Then we have the steps 2 through 4, which are mostly bioinformatical work, and at the end come up with some sequence data and information about genomic alterations. And these genomic alterations have to be then interpreted in the step 5 to know what those clinical consequences of those alterations might be. And this is still manual work to a large degree to be ultimately used and applied in clinic, which is step 6. So doing that, we actually come up with a group of diverse cancers that all have very unique molecular profiles, since ultimately actually every cancer is unique. And what we then do is try to find alterations that also serve as predictive biomarkers for a targeted therapy. And if we actually find a patient where we have a possible therapy to target that biomarker, this usually works great, and we really have miracle stories sometimes, like these pictures I can show you from our clinic, from a patient with a RET fusion positive lung cancer who had a dramatic response to a RET specific inhibitor. And this is just to show you that precision oncology in general works very well for a lot of patients. And even though I'm going to be a little bit more critical of what we do, I just want to give you the basic idea that we do have those stories, and I'm sure that Razelle Kurzrock, who is going to be our keynote speaker later, will give you some more of those examples. However, I also want to show you some criticism of what we are doing at the moment. Where do we stand in precision oncology? We recently summarized a lot of precision oncology trials, most of them retrospective trials, in an advanced cancer population. Please keep that in mind. And if you look at the median across all of those studies, we are at the moment treating about 1/3 of those patients that enter precision oncology programs, and about another 1/3 of those patients actually have either an objective response or disease stabilization. That means that we currently are able to offer drugs to some of the patients where we find predictive biomarkers, that we still have a large group of patients where we either can't offer anything or where what we offer doesn't really bring them a benefit, meaning that we are actually pretty good at the moment at finding the right patient for a given drug, but we are not yet very good at finding a drug for every patient. And I think that is something where we still need to work on the field, and I just want to give you some ideas now of where I think the field might be going or what we are able to improve as of now and what we are working on. Naturally, the development of novel drugs and novel drug combinations is something that comes into mind that I'm not going to tap on today, because that is very complex work that is done by a lot of pharmaceutical companies. But I'll talk about 2 things that I think are being done in clinical routine and that we are doing research on, and lots of other people as well. And that is, first of all, the clinical interpretation of molecular alterations. Because as I showed to you earlier on slide 5 of this figure, the interpretation of genomic alterations that come from high-throughput sequencing are still largely manual work. That means if I present a physician with a list of molecular alterations, it is not clear if he's going to find the same evidence that another physician finds. And in fact, we did this with a round-robin test. So we created fictional patients and sent them to molecular tumor boards across the world. And this is one example of a patient with a lung adenocarcinoma with a KRAS mutation and some other mutations. And we actually sent this fictional case to about 35 tumor boards and got answers from 6. And basically, to keep the long story short, it is pretty heterogeneous what tumor boards recommend. And that is something that shouldn't make us frown, but rather see that we are talking about patients that have a very long history and very difficult to treat cancers, and where we oftentimes do not really have very good options, but also that we need to improve ourselves in where do we reproducibly get the evidence. Talking about KRAS mutant lung cancer, I also just want to briefly show you how dynamic the field is at the moment, because yesterday the FDA approved the first KRAS-specific inhibitor, not for exactly this KRAS mutation, but for another KRAS mutation. And I just want to show you that the field of precision oncology is really moving very quickly, and we start getting novel therapies by the month, and which is really great for our patients. However, we need to work on the reproducibility of our recommendations, and for this, an international collaboration worked on developing databases where we could actually put in and store data on drug-gene interactions to enable us to actually find predictive information of those on those alterations that we find in our tumors or in our patients' tumors. Those are 2 links to those databases that are copied. They are open source and free to use, so please feel free to contribute. We also tried to look into the performance of tumor boards when they are faced with very complex data, but also use those databases and try to use structured workflows. And we did this with a cohort of about 45 patients that underwent extensive sequencing, and we compared the recommendations for both biomarkers and therapy options from the Heidelberg and Berlin molecular tumor boards. And those are the results of this comparison between the two tumor boards that I'm showing you. So this figure is actually the agreement rate between the two tumor boards on the x-axis for the biomarkers and on the y-axis for the therapy options. And we can see that if we both work with the same structured workflow, we actually get much better in agreement rate for those patients. So every patient is represented by a gray dot. And we are actually able to achieve a median treatment recommendation overlap of about 75% between both tumor boards, which is actually, I think, pretty good, given that those patients have very high dimensional data with more than 200 aberrations per patient. And this is also relevant for the clinic because those patients where we are able to find reproducible information and give them reproducible treatment recommendations also have a better outcome when treated with targeted therapy. So the green line is those patients that received a targeted treatment that was based on a concordant treatment recommendation by both tumor boards, whereas those patients that received either a differing or that followed either a differing recommendation or received a physician's choice therapy had significantly worse outcome. And these findings mirror those findings that have been published previously in the landmark trial led by Razelle Kurzrock, who will speak later today, published in 2019, where they showed that those patients that receive targeted therapy, which is well matched to the molecular profile of the patient, actually have a better outcome than those patients that receive targeted treatment that is not very well matched to the tumor, which is another call for individualized therapy and also for combination therapy. The second thing is understanding tumor biology, because with a clinical interpretation, we are only able to go as far as evidence exists. But we are creating a lot of new knowledge, and we need to use this knowledge to really understand what the tumor does and where it goes to be able to follow it therapeutically. To give you another example of a patient with an advanced breast cancer, this patient was found to have lots of mutations, which are here on the right side of the slide. Most of them are very well known, including estrogen receptor mutations, PIK3CA mutations, also a RET fusion. We actually have lots of drugs for those alterations, but if we look at the tumor in bulk sequencing, we really don't know what each and individual cell does, and if all of those mutations are together in the same cells, if some of them are actually following others. So we actually need to look more into the tumor in detail, and this is work that is done by the Tirosh lab, who just gave his amazing talk, because he really has been at the forefront of RNA sequencing in cancer and made us understand in detail how different the tumor works within itself. And if we actually look at the different tumor populations, they also differ in sensitivity to various treatments, as shown in this great paper. And therefore, I think it's not enough to just look at the evidence, but also to understand in detail what the intra-tumor heterogeneity has as consequences for the patient. And we are still not finished with this, because the tumor is also a moving target that changes as we speak and as we treat it. And therefore, we still need to also include temporal information, not just the spatial information as done here, because if we look at the same patient, on the left-hand side, you see the first sequencing about 2 years ago. The right-hand side is the most recent sequencing. You can see that the tumor actually changes the types of mutations it has. So it's really important to also follow the tumor therapeutically to be able to understand its individual biology. Having said that, I want to come to a summary. I believe that with new drugs and new drug combinations, we are actually moving forward in this exciting field. I believe that we still need to focus on the annotation and clinical interpretation of molecular alterations, because this is a complex task. I believe that the integration of novel sequencing technologies like RNAs or single-cell sequencing as landmarked by Itay Tirosh is really something that might help us greatly, but we also need to integrate a temporal resolution to make sure that we follow the tumor as we treat it. With that, I want to thank lots of people. So first of all, our 3 guest speakers, who we're very grateful to for being here today. I want to thank the team at the Comprehensive Cancer Center, together with Professor Keilholz, who is also my mentor in the clinician scientist program, and lots of other people from the clinic, including Sebastian Ochsenreiter, my clinical mentor, the people in Heidelberg, the bioinformaticians, also the VIC consortium internationally, and of course the clinician scientist program for organizing this thing. And last but not least, I want to thank Dushka Dragoon, who did an amazing job and will not be forgotten. Thank you.
Thank you so much. This was another very interesting talk, and both talks together show the complexity of cancer and the complexity of how to tackle cancer. And this is really of high interest. I see one question in the chat at this moment, so please feel free to add your questions. Before that, I will ask my personal question to Itay Tirosh. You mentioned the interaction between glioblastoma cells and macrophages. And my question would be, is this a special subtype of macrophages? Because macrophages come in so many different flavors that is required to cause this interaction, or is this something that is a reaction of macrophages to glioblastoma so that they will just move to a certain state, cellular state, in order to have this interaction?
Yeah, so the specific interaction I was talking about is mediated through secretion of oncostatin M, and oncostatin M is secreted by macrophages and microglia more or less regardless of their diversity. So more or less all of the macrophages in the glioma environment secrete this, but we do see that there's a higher secretion of OSM by the macrophages in a glioma compared to the macrophages in the normal brain, or specifically the microglia. And so one important axis of variation here is between the microglia that are resident in the brain and the macrophages that enter the glioma through the circulation. But again, both of them secrete OSM, and so both of them are responsible for this interaction.
Mm-hmm. And if you look into glioma, is there a predominance of a certain subtype of macrophages, or are they displaying just the diversity we know from macrophages?
So there's definitely diversity. It's difficult to put the threshold and say, when do you turn from one type to another type? There's definitely a lot of diversity. We also see changes that correlate with progression of the glioma. So as the glioma progresses, we see a gradual shift from the microglia towards more macrophage cells. And there's additional axes, but it's difficult to say exactly what's the amount of diversity and which types dominate.
Okay. Then the question from the chat also to you. The question is whether those different glioblastoma cells you can see with your method, whether they are differentially sensitive to chemotherapy agents.
Yeah, so we are just now trying to, we're just now trying to test exactly that in two different ways. One way is to compare patient samples at diagnosis before receiving any treatment and the samples after relapse. And so we are comparing matched samples on the same patient and looking for changes. We do see changes. We do see that some states become more common than others, but this is different between different patients. There is no global shift that we can expect a priori. So it doesn't seem like there's one state that is particularly sensitive or the other way around. We are also trying to test that in the lab. So we are trying to identify model systems that would recapitulate the patterns that we see in the tumors. And then using those model systems, we are directly screening them for drugs to see whether there are particular drugs that affect one state more than the other. We definitely see differences, but these are not really the same drugs that are being used clinically. So the relevance for that question is not entirely clear at this point. What we would like to find are drugs that are particularly useful in eliminating each one of those states.
I have a question. I'm not sure if I'm online.
Yes.
Yes, also to Itay Tirosh. So just a quite general, maybe provocative question. Are you optimistic that the recurrency of those heterogenic states within the same tumor entity is something that can be treatable? Or are you more pessimistic and think that the heterogeneity is something with those fluent states that is going to be very different to track therapeutically?
So I guess I would say that because we see the heterogeneity remaining in the relapse and we never see one state that is completely lost, we tend to ascribe that to plasticity, that regardless of whether you were specifically efficient in eliminating one state, if the states can transition from one to the other, then at relapse you will see all of them again. So that is the problem with looking at the relapse and trying to assess which states are the more drug-resistant ones.
So the next question from the chat is a more general question to both of you. I will just read it for you. We have the heterogeneity of the tumor, which is very complex, thanks to the two great talks. How do you integrate the distinct risk profile of the individual patient?
Maybe I'll start first. So yeah, this is a good question, and I think this brings me to something that as a clinician I think is very important, because when we talk about genomics and intratumor heterogeneity, we tend to pretend like this is the only information we have about a patient, but that's not true. In clinic, we do precision oncology all the time because every patient has a different frailty score, every patient has a different age, every patient has different medical conditions. So that means we really have to look at the individual patient's risk scores for many different things. And I think sequencing can only be one part of this. I'm not sure if I've answered your question, but that is my thoughts on that.
So maybe I can add just one more point, which I also try to make in the talk itself, which is that certain things are very patient-specific, and then you have to consider that on a patient-by-patient basis. What we are trying to highlight are the aspects of diversity that are more common across different patients. And then if you find those things, you don't really have to think about this for every patient individually. The goal is more clear, because we know that, for example, in glioblastoma, we should expect to see those 4 states. So now we can more easily incorporate that in how we think about therapy.
So the last question to both of you, or either of you, where do you think is the role of single-cell analytics in precision oncology, in clinical precision oncology? Is there a role, or is there an emerging role?
So maybe I will start this one. I think that this is more of a research tool at this point, and maybe in the future it will have a role in the clinics, but I think it's a little bit complex to generate this data, good data, and to then analyze it properly. And I don't think it points very, very clearly into a therapeutic strategy uniquely for a single patient. So I think that this is more of a research tool at this point.
I think, or I believe that it has a role in precision oncology, and that it will move into clinical routine, maybe not just by itself, but together with bulk sequencing and maybe targeted sequencing with a high depth and very high confidence of those genes that we are interested in. But I think together with those sequencing tools, I think single-cell sequencing will help us greatly, and I'm quite positive that we'll be able to integrate this.
So a question we should revisit 5 years from today, I guess. Okay, thank you both of you for this exciting talks and the exciting discussion. And now it's my great pleasure to introduce a key driver of precision oncology, that is Razelle Kurzrock. Razelle Kurzrock is a medical oncologist, and she has had a long period at the MD Anderson Cancer Center, where she developed the large phase 1 clinical trial programs. And then she recently moved to the University of California in San Diego, to the Moores Cancer Center, to continue precision oncology there. And I'm looking forward to her presentation on her view of current precision oncology.
Are the slides working?
Your microphone is still muted.
Hello?
Yes, Ms. Kurzrock.
Yeah.
Can you see the slides or you can't?
No, not yet. If you can start your sharing again. No, not yet.
Not sure why it shares.
You can also just start your presentation. We can play it from here. Now it works. We have it. Just start it. And then we should be good.
You're able to see it right
Exactly,
now?
exactly. Now everything is good. You can start.
Okay. I just need to, okay. So it's full screen now, is that correct? Can you see it? Hello? I cannot hear you. Are you able to see this? Hi, this is Dr. Kurzrock. Are you able to see the screen? Okay, so you're all good, right? And you can hear me, is that, okay, great. It looks a little funny here, but as long as you're good, and just let me know if something happens that isn't working. I just, and are the slides moving now? Are you able to hear me? Okay. So thank you so much for having me here. And I'm going to talk about precision medicine and how the future is here. And I've really enjoyed some of the prior talks. And I hope a little bit what I'm going to say is going to expand on them as well. And so again, thank you for the honor of having me here. This is my list of disclosures. And then I'd just like to give you a little bit of background. So my interest in precision medicine and my experience is derived from my experience at two institutions. First, MD Anderson Cancer Center, where I founded and built what really became the largest clinical trials department, probably worldwide. And then University of California, San Diego, where I led the precision medicine program. And what we discovered, MD Anderson was first. We began to use precision medicine back in 2007. The technology was really primitive then. We had 12 genes, we had some hotspots, and we tried to match patients with drugs. And then we began to get a glimpse that maybe this could actually work for our patients. But then as the years went by, around 2012, next generation sequencing became available for our patients. And we were really, really excited. This was tremendously powerful technology. We had so much more information. And then we realized that this actually presented a very big problem to us because we had so much information now, and it was turning out that tumors were extraordinarily complex, and they were each different from each other. And the way that I had done oncology throughout my lifetime, and the way everybody was doing oncology, the science, the sequencing was telling us that it wasn't a really good fit. And that every tumor was complicated, every tumor was different from each other, and that we needed individualized combinations of therapy for each patient. And of course, that's not the way we do things. So this really required almost a deconstruction and rebuilding of the entire field on how we treat patients to fit the precision medicine realm. And I want to say that that has been, I think, the biggest challenge. There's, of course, challenges with understanding variants and so forth, but it's actually operationalizing precision oncology. It's actually getting the drugs to patients. It's actually doing customized combinations, which is even the bigger challenge, I believe. So my take-home points is that precision oncology is all about getting the right drug to the right patient at the right time. And that, first of all, every metastatic tumor is unique and complicated, like malignant snowflakes. I'm in San Diego before that, in California. Before that, I was in Texas, nice and warm, but I grew up in Canada. And so I know a little bit about snowflakes. And I think they're a really good comparison to cancers, what we know now. Each snowflake, the crystal structure is unique, and each one is very complicated. And that's true for metastatic tumors. The corollary to that is that each patient needs customized, individualized combination treatments, not single agents. And we've been going with a lot of single agents in precision oncology, and I think that's limited us. The number three is that the pillars of precision oncology are genomics and immunotherapy. They are not separate. They're actually married to each other. And targeted therapy, genomically targeted therapy may be best suited for simple tumors, while immunotherapy may be best suited for genomically complex tumors. So the whole field has been driven by what is absolutely breathtaking moves forward. The first human genome was sequenced about 2003. Actually, in the 1990s, everybody said that it would never be sequenced. Anybody who thought the human genome could be sequenced was working in science fiction. But by 2003, we could sequence it, but it took 13 years for one genome. Today, we can get results back in 1 year, in 1 day. It took $2.7 billion for one genome. By 2007, it was down to about 100 million, still a little bit high for most insurance and for my research grants, but now we can get a genome for $1,000. And so this is really breathtaking progress, almost unparalleled in human history. And really, it's what's fueled the precision medicine, what I think is transformation or even revolution. So a whole new era was born. And this is the era of tissue agnostic approvals driven by genomics. And the first one was pembrolizumab, which is a checkpoint inhibitor, which was approved for solid tumors based on mismatch repair gene defects. It was approved on May 23rd, 2017. And there were three aspects of this approval, which are completely transformational. First of all, it was the first tissue agnostic approval, meaning it was approved for all solid tumors, wasn't based on a histology. Second of all, it was the first approval based on a genomic marker for a tissue agnostic approval. And then the third part of the approval, I think, is actually the most revolutionary, but it's the part that maybe people know the least about. In part, it was approved based on retrospective real-world data, so not just prospective data. And since then, the FDA has approved other drugs entirely on real-world data. And I can't overemphasize how that is absolutely transformative. If we can use real-world data, millions of data points that are in our computer bases now for approvals without doing a 5-year trial. But that's not the end. That was just the beginning. So since then we've had more agnostic approvals. We've had larotrectinib for NTRK fusion adult and pediatric tumors. We've had entrectinib for NTRK fusion adult and pediatric tumors. And we have pembrolizumab again, this time for high tumor mutational burden, just approved about 9 months ago. And so that brings me to the part that I wanted to say about the marriage between genomics and immunotherapy. And people think of these aspects of treatment as separate. Genomics, immunotherapy, they're separate fields. They're not separate fields. They're a couple and they're married to each other. And in what way does that occur? Well, the way immunotherapy works, checkpoint blockade in particular, is that the tumor puts the immune system to sleep. The immunotherapy wakes up the immune system. But the immune system still has to recognize the tumor. And the way it recognizes the tumor is by how many mutations are there, because the immune system is set up to look for things that are different than normal. And the more different it is, the more easily it is to recognize. And so these tumors that we once thought were completely untreatable, even I thought that they were untreatable, that were just full of mutations, a total mess. These are now the best tumors. These are the patients where you see the long-term complete remissions that we never dreamed would happen before. So we had a molecular tumor board at UCSD. We've had really a lot of experience with molecular typing, looked at about 21,000 patients, not at the tumor board, but we've done about 21,000 patients with clinical grade typing. And we bring together experts from many different fields of medicine, but also bioinformaticians and our basic scientists. And I think this is really important. Number one, not just the quality of the panels, and you have to have a really good quality panel, next generation sequencing, but the quality and the experience and the diversity, multidisciplinary nature of the molecular tumor board. And we actually recently published our experience with 715 patients in Nature Communications. And so I'm going to give you some patients that came from my own clinics, and to give you examples of what we do. And so this is a patient that I saw in January 2016. 49-year-old woman. She had come from San Diego to Saudi Arabia. She was in a horrible shape. She had a high-grade neuroendocrine cervical cancer. She had waited too long. She'd had surgery, radiation therapy, and chemotherapy. And she had an enormous abdominal tumor. She looked like she was 7 months pregnant, but it was all cancer. Her pending bowel obstruction, her kidneys were about to shut down. And we had a big problem. We didn't have time to get a tissue biopsy and do NGS. So what did we do? We did a liquid biopsy. We had a huge liquid biopsy program. We've done almost 6,000 patient samples. And so we took a blood sample and we sent it off for genomics. And of course, that's much faster. We were in a hurry. And what we found was this. Her circulating tumor DNA was just full of alterations. The usual patient has 3 or 4. She had over 20 alterations. So we took a guess and we said, she has a high tumor mutational burden. And that wasn't really known at that time, but we thought maybe she has hypermutated ctDNA. She has a few days left to live. We have one shot at this. She went on our protocol, which allows matching of patients to the right therapy, or what we think is the right therapy. And so she got nivolumab, which is a checkpoint inhibitor. This is her tumor before. This is 8 months later. 3.5, she did go back to Saudi Arabia after 3.5 years. That was my last contact with her. She was still in complete remission. So I think it shows the power of matching patients, but also the power of new technologies. In the meantime, while we were treating her, we did do a tissue biopsy, and it turned out that we were correct based on the blood. She was full of mutations. Tissue usually has 3 or 4 mutations. She had a high mutational burden of 53. She had mismatch repair gene defect. And important point here. Cutting edge technologies are needed for complicated cancers. If we had had just tissue, we could not have done this for her. She just really didn't have long enough to live. So liquid biopsies are important. We now know you can do tumor mutational burden, and microsatellite status is now available on these tissue biopsies. So We're all very excited about immunotherapy, but I want to show you that it's not all good, and genomics can help us even with the problems. So we and others have defined what we call hyperprogressors in immunotherapy. We first saw this in our clinic a few years ago, and we published on it. And these are patients that you give them checkpoint blockade, and it looks like you poured gasoline on a fire. Their tumors explode. It really accelerates in a dramatic fashion. This is an example of a patient, breast cancer. She received nivolumab. This is 2 months before nivolumab. This is right before nivolumab. And you can see there's not much of a change, but she gets nivolumab, and 6 weeks later, her tumor just explodes. And she has an MDM2 amplification. And when we looked at our patient population, we found that there were 2 alterations that were associated with a higher risk of hyperprogression. This doesn't mean that everybody with these alterations hyperprogresses, and it doesn't mean that that you can't have hyperprogression if you don't have these alterations. The story is just beginning. But number one is EGFR alterations, which are known to be associated with resistance, but some of them also seem to be associated with hyperprogression, and an especially high risk for patients with MDM2 amplification. I'm a scientist. I don't know why. So if you ask me why, we don't understand it. We've had some of our scientists looking at it, but the association seems very high. So here's an important point. Next generation sequencing can help define not only who to treat, but who not to treat. And that's probably just as important. So let's move on to the first thing that I told you about, because I think it's the most important thing. What if every patient with metastatic disease is different, just like snowflakes? Look at those snowflakes. Every crystal structure is different. Everyone is very complicated. And this is just next generation sequencing that we did. This is actually Foundation Medicine from breast cancer. We've done thousands of patients, but I just put this on the slide just to give you an example. So these 2 patients both have ERB2 amplification. We all know you treat that with HER2 targeted agents, but look at the rest of their profile. They each have multiple other alterations that are relevant, and the alterations are not the same. And yet what we do is we treat patients the same just because they have something in common. That can't make sense. If you just look at this, you can just see that it cannot make sense to give patients that are so different the same regimen just because they have something in common. So we started a trial. We actually started this back in 2015, and that's called iPredict, Investigation of Profile-Related Evidence Determining Individualized Cancer Therapy. And what we wanted to do was customize combinations. And a second part of the trial was actually to bring the drugs earlier to newly diagnosed patients. The first part, there's a publication in Nature Medicine, so I'll talk about it. And we published it in April 2019. And in that trial, we gave customized combinations to each patient, and we were able to match about 49% of patients. That's a very high matching rate. A lot of trials are anywhere from 5 to 25% of patients. So I want to reemphasize, when people talk about the scientific barriers for precision medicine, but the biggest barriers are operational. It's about getting the drugs. And we found a way to get the drugs to the patients. And then our IRB allowed us to do customized combinations. So this is from the Nature Medicine paper. You can see 45% response rate in the high match patients, 16% in the low match patients. So it's not about, is a patient matched, yes or no? It's about the degree of matching. And so low match patients included a small number that were unmatched, but also patients that had a low matching score. Just to give you an idea, let's say there's 10 alterations. If you match one of them, the matching score is 10%. But if you have 2 alterations and you match one of them, the matching score is 50%. So the importance here was trying to get customized combinations. And here's the survival for high match patients, about 60% at 2 years, low match patients, about 20% at 2 years, a very dramatic difference. And here is an example of a patient. I'll just give you, this is what we do did routinely for a patient, but this is an example. This is a 62-year-old man with a poorly differentiated carcinoma of unknown primary. He had a KRAS mutation. It's an unusual mutation. So we had our scientists look into it. It is activating. So we used a MEK inhibitor. We used specifically trametinib. He also had an ARID1A alteration. ARID1A is a chromatin, part of the chromatin remodeling complex, does a lot of different things. But one of the things it can do is sensitize to PARP inhibitors. So he got olaparib. So he got trametinib with olaparib. This is a customized combination, had a high matching score. And you can see the very nice, remarkable response that he's had, and therapy is ongoing at 15 plus months. The last thing I wanted to mention is my work with the Worldwide Innovative Network for Personalized Cancer Therapy, which is an organization that is devoted to the global delivery of precision medicine, and also to the concept that genomics, which we've really emphasized, is just the tip of the iceberg. We need to be looking at transcriptomics, and then beyond that at proteomics and epigenetic changes. And together with international colleagues, we did the first trial from the organization. It's a signature trial called Winter. And what we did there is it's the first precision medicine trial for solid tumors that I know of that included both transcriptomics and genomics for solid tumors. And we found that the transcriptomics was indeed an important addition. And the results of that trial were published in Nature Medicine. So I would just want to finish off by saying that the genomic immunotherapy era will revolutionize not just cancer medicine. Cancer is at the forefront, but I think precision medicine is going to influence all of medicine. And here is something that I find fascinating personally. Bladder cancer has common FGFR3 alterations, and we have inhibitors that can inhibit it and get responses in bladder cancer. But to me, fascinatingly, achondroplastic dwarfism, which is one of the most common types of dwarfism, has the exact same mutation, not just the same mutation in the same gene, FGFR3, but actually the same locus. And that brings up questions like, why don't these children get cancer at an increased rate? And we don't understand the compensatory mechanisms. And then even more interestingly, could we use our FGFR inhibitors and actually change the phenotype of these children? And I think that's a fascinating question. It's actually been asked in animals, and the answer is yes, you can do this successfully in animals. And obviously nobody's done it in patients yet. And then I just want to finish by thanking the precision medicine team and the patients that contributed to this. So I'll end there and thank you.
Thank you so much. This was a fantastic talk, and obviously many reasons to discuss. It's too bad that we don't have a face-to-face meeting. It would be nice to have a chat for at least 2 hours. Please put your questions in the chat and I will read them for you. My first question is something I'm intrigued with. You mentioned the integration of immunotherapy and molecular targeted therapy, which we also follow at the Charité, and I think this is important. But the question is whether PD-1 agents are not just the beginning, whether we are moving into something that is called precision immunotherapy also, to look for the reasons how a tumor got resistant to the immune system, the escape mechanisms, in order to come up with combination immunotherapies or combination molecular target and immunotherapies on a more rational level than we do today. What do you think about this area?
I think you've totally hit it. The issues with immunotherapy is great for about 15 or 20% of patients. And I think it was hanging fruit for the anti-PD-1, PD-L1 agents. I think for the other agents, we just have not learned the lesson we learned with genomics, and we need to learn it. We are giving all sorts of different immunotherapy agents. I think in the United States, you need about 400,000 patients per year to fill all the clinical trials, and almost none of them have biomarkers. This is a recipe for failure and lack of optimization. So there are now really good assays. We've been using an assay called OmniSeq that can look at the immune profile of every patient. And I absolutely agree with what you said. Just like you cannot give an ALK inhibitor to a patient that doesn't have ALK rearrangement, expect a response. I don't think you can give immunotherapy blindly, which is what we're doing for the most part, and expect a response or an optimal response. We got a little lucky with the anti-PD-1 agents, so there's a fraction of patients that just respond. And now we're looking at their biomarkers, but absolutely. In fact, one of the things that I think is very sad is drugs that are being abandoned, like IDO inhibitors. And IDO is elevated in about 5% of patients. So of course it doesn't work because it's only a small fraction of patients and we're not selecting them. So I absolutely agree with the precision immunotherapy concept that you brought up. It's the way to go.
Okay, thank you. Now the questions come flying in. The next one would be, how do you think high matching rates can be achieved with complete omics data rather than current panel sequencing?
I think they can be achieved in the same way. We need to understand what the important drivers are. And then the issue, the bigger issue, even bigger than understanding, is two issues. Number one issue is that you have to have the drugs. And you have to have access to the drugs. And at UCSD, we found a peculiar way to get access in the drugs. I've learned now working with WIN and other organizations that every country is different. In some countries, there's ways to get access, and in other countries, very difficult, especially off-label drugs. So the first thing is, can there be a way to get access to the drugs? And again, we found a way. I'm not going to go into it, but I joke that we hired a magician, but we found a way to get access to almost any drug. And then the second thing that has to happen, very, very important, and again, this is the next step after you have an understanding of the omics, is you have to be willing to do customized combinations of drugs. Most doctors, including me, I'm actually a phase I expert, have been taught that you can't do a combination if it hasn't been tried in a phase I trial. And with our publications now, the 2 publications in Nature, and we're soon going to have some others that come out, we showed that you can do de novo combinations safely, that this is a rule that we imposed on ourselves. But by lowering the dose and then titrating each patient up when phase I hasn't been done, our data safety monitoring board showed that we can absolutely do this safely. This is critical to get high matching score, that you be willing to combine drugs that have not yet had phase I studies.
The next 2 questions are exactly in the same direction as you just discussed, and I will read both of them because I think you can best answer them in combination, because they are on combinations. How do you think will regulatory authorities have to deal with customized combination therapies? And have there been reports of clinically significant drug interactions in patients who received combination therapies?
Right. So I think it's, I'm going to tell you a little story because I want to tell you how I came to this. Remember, I led one of the largest, if not the largest, phase I departments when I was at MD Anderson. So I didn't believe we could do customized combinations either. I've been taught that just like everybody else. So I'm going to tell you a little personal story of how I came to this. So I had a friend who was a physician, and her husband was a physician, and our children grew up together, and she had metastatic breast cancer. And after I left MD Anderson in Texas and came to California, her disease recurred. She had been sick for a long period of time, but now she had a very serious recurrence, and she wrote to me, and she was on TDM1, which is a HER2 inhibitor, but she had a PIK3CA mutation. So I suggested that maybe everolimus should be added, and her doctor said, you cannot add everolimus because nobody's ever given everolimus with TDM1, the HER2 targeting agent. And she wrote me an email that actually for me was the birth of the iPredict study. So remember, she's a physician, but she's not an oncologist. She's a different type of physician, and her husband's a physician. So she hadn't been trained in our thinking. And she wrote me an email, and she listed all the drugs that she was on because she had a lot of comorbidities. And she was on about 15 different drugs for all her other problems that she developed. And then she had in blue the TDM1. And she said, the title of the email was, "Where is the safety data?" And so what she was saying is, I'm on 15 different drugs and nobody complains, but you want to add everolimus and all of a sudden nobody can do it. So I realized that at that moment that as physicians, we in the United States and really in every country, we individualize therapy, we give customized combinations, we do it even without thinking. The average cancer patient comes into the cancer clinic on 8 different drugs that are unrelated to their cancer. It treats their depression and their infections and their arthritis and their diabetes and their heart disease, whatever other conditions they have. Nobody even thinks about it. But if we want to do 2 drugs that are cancer related, all of a sudden everybody gets excited. And I think that's a legacy of the cytotoxic era, because these drugs, the cytotoxic drugs, were very toxic, and putting them together was dangerous. But we have much easier drugs now. We learned how to put them together very safely. And I think that it is something new for the regulatory agencies, but there probably is no other way around it. Because if we have 300 drugs in oncology, if you look at 3 drug combinations, about 4.5 million, we're never going to do all those phase 1 studies. We're going to have to figure out how to do it safely. We've already published on this, but I think that's the future. De novo combinations done safely with certain algorithms.
I fully agree, and I know that this is a big headache for the regulators, but they simply have to move with us in order to really move forward and allow what is reasonable. And of course, we will have some failures here and there, but again, we have to learn from them.
Right. So I want to say that, yeah, so one of the purposes of our publications, we have the Nature Medicine, we have our Molecular Tumor Board, Nature Communications. We have another paper that is very close to acceptance in a major journal. So I think we've sort of set the path there. We need other groups to do the same studies.
Okay. So the last question is also a tough one. Do you recommend to combine solid and liquid biopsy sequencing in order to find all druggable alterations?
The answer, thank you for the excellent question. The answer is absolutely yes. So we do that routinely. We do the tissue. Often the panels are larger, but we do liquid biopsies at the same time. And liquid biopsies reflect shed DNA from multiple sites. So, and we look at the results from both biopsies, both the blood biopsy and the tissue biopsy at the same time. And that helps with the heterogeneity and all the other issues. These are complementary techniques.
Okay, unfortunately, we don't have time enough to go into details of this question because it's a very interesting one to be followed up at another circumstance. I thank you so much again for this great talk and the excellent discussion. And now it's time for me to turn back the microphone to the to get to
me, Britta Siegmund. Thanks,
Britta
Uli.
Siegmund.
Thanks, Uli.
For the closing remarks of this exciting symposium.
Excellent. Thank you, Uli. Yes, I'd like to take the opportunity in the end and shortly summarize our last 2 days and the 10th year's anniversary of the BRH Charité Clinician Scientist program. We started yesterday morning with minor technical problems, which we ignored at the end and proceeded. And to all of you who didn't really listen entirely to the first session, we will make the first session available and you'll be able to listen to the first session. But the session was dedicated to Dushka, and I'd like to transfer here the message of her patients, of her parents and the friends of Iga and Tanya, to whom I talked yesterday evening, and both of them expressed their great gratitude for the first session. I think this is very important for us and for them. We then moved on to the political session and highlighted, and I think this was very successful, that we highlighted what the significance of this program is for the Charité, but also for the University Medicine in Germany. And I think in particular the second part, the results of the evaluation program, really highlighted this point and also highlighted for us, for the board, on which parts we have to work on. I think this is also very, very important. Then we moved on to the scientific sessions. We started yesterday afternoon with the digital session. We continued this morning with the metabolic vascular session, continued with neuroscience, and ended now with the oncology session. And I don't want to go into content, but I think we had really excellent international speakers and national speakers. But I also really like to express my thanks and my admiration to all our clinician scientists and junior clinician scientists who gave excellent talks and contributed in the discussions. And I think these were wonderful tandems, and I really enjoyed all 4 sessions, although some of them were really far away from my original experience. So I think we had a great science over yesterday afternoon and today. Last but not least, I'd like to express my thanks not only to all speakers and junior and clinician scientists and to all board members and participants of those 2 days, but I also like to express again my thanks to the team, to the BR team, who really made all this possible, and who I think if we would be here in a room, this would be a great applause right now, which we can't. Yes, we're trying to do this. And thanks to the technique for the help here. And I think this is really, without them, we wouldn't have been able to do this. So really, thank you. Last, but also important, because tomorrow the sun is going to shine in Berlin, I wish you all a wonderful evening and a great remaining weekend. Thank you.